And I'm not talking about revolution. This is a scene from, or at least a depiction of Russian Revolution, the insult or the assault on the Winter Palace in St. Petersburg that happened just about 100 years ago. I'm talking about revolution that is going to have a lot more profound consequences than even Russian Revolution in the long time of humankind. This is a revolution that has to do with our ability to engineer biology. And that's what I'm going to talk about today. That is what has happened in the last couple of decades, but what is increasingly happening these days in our change of in the change of how we deal with biology, right? So for the most part, biology has been analytical science. We've been studying how things are. Well, at this point, we can make things. And that is both exciting and kind of scary. So I'd like to give you some ideas of where we are, what we do in general as synthetic biologists, as biological engineers. And that will be the first half of my talk. And then I'll tell you what we do in my lab along these lines. Now, I don't like monologues very much, so by all means, if you have questions, raise your hand and do feel free to interrupt me. So, let me give you an analogy here. So if you think about sort of progress in, say, engineering of any kind, let's take architectural engineering. Right? So that's where we started as humans, right? Building those huts with bones and skin of animals, or with stones, or just living in caves. Well, at some point we realized that there are actually blocks that one can use, and so with bricks and some glue in between, one could build really nice palaces and bridges. And these days, we don't really use much of a natural components. We use a lot of synthetic components to build buildings like this new World Trade Center, or this building in Dubai. So basically, what let us transition from this stage to this? And I would argue that there are just a few critical things that allow any kind of engineering. One needs to know their parts. One needs to have tools to put these parts together. One needs to understand the rules by which these parts need to be put together. And one needs to design a specific language. And by language, I mean the language of any particular discipline. Right? So parts in architecture would be, I don't know, bricks, concrete panels, right? Tools, cranes, perhaps. Rules, well, one needs to know physics, right? Otherwise, those buildings will crumble. And one needs to design, when one is talking about buildings like these, one needs to have a pretty sophisticated computer software to just handle all the small parts. Well, what I'm trying to convince you is that biology is entering the stage where we know what the parts are. We have just gotten the tools to play with them very precisely. We are learning very quickly what the rules are. And so we should be able to engineer things, and we are engineering new organisms. So this is what I'm going to tell you about a little bit. So what are the parts? Well, this is a representation of human genome. These are 23 pairs of human chromosomes. We know that all of these chromosomes contain genes, and these are units of life. We have learned how to sequence these genes, that is to read this code of life. And it's a pretty simple code. On the surface of it, it's just A's and T's and G's and C's. It's just that one needs to understand that language, and fortunately we do. We do understand this language to the point to break this text into parts. We know how to identify a part of this string of letters that would encode a protein. We know where so-called ribosomes would bind to make that protein. We understand how this gene can be expressed or made into a functional unit. And so we can formalize this knowledge in these various parts and components. We've had these tools to cut DNA for quite some time, and we've been playing with those tools. And in fact, we've been not only playing with tools in cutting, we've been playing with tools of inserting genes in the existing strings of DNA. We've been doing it very efficiently with bacteria and other microbes. In the last few years, the tools to do these types of precise engineering in mammals have emerged. And so we have these tools now. We can precisely splice in one of these chromosomes and cut out a gene, or add a new gene, or change that perhaps mutated gene into a normal one. That's really very exciting. Where do all the parts come from? And what is, I guess, the realm of parts that one can play with? Well, since all life forms have the same type of code and essentially the same parts, it turns out that there is no limit. Any genome has genes that can be put into any other organism. So this is what is known as tree of life. If we look at, and so since we've been reading the code of life for a long time, we've been sequencing these genomes of mammals and humans, but mostly microbes. Because if you look at this tree of life, it doesn't look like a tree really, but that's just a historic term, I guess. So each of these branches represents a major clade of life. All of these are bacteria. Believe it or not, we, humans and plants, belong to a tiny little branch on one of those trees that belongs to archaea. So we share ancestry with these tiny little creatures. And we know that because we have read the code of life, and that's what it tells us. We come from the same lineage. But each of these bugs, so to speak, have genes, and we can take any genes and put them in any other organism. People who are really at the front edge of this, like a colleague of mine, Chris Voigt at MIT, they have actually designed a programming language for synthesizing living cells. Right? So this is a program. This is what you'll see as what you'll have a computer program for designing, I don't know, a table, one of those buildings, or a computer. This is for a living cell. So it has these strings of letters. This is a gene. That's how you put this gene in. And this is a code in between these lines. What you do with this gene, you take this gene out, you put it in here, you put this regulatory elements to control how much it's expressed. And what is really cool is that it works on a relatively small scale, but it works remarkably well. So we understand at least some parts of biology, some parts of rules in biology well enough to program cellular behavior, then analyze it, and realize that our programs work fine. And so these are some of the schemes that one uses. And if there are electrical engineers in the audience, they'd recognize, I mean, this looks like electric schemes. And in fact, that's the background of Chris and a bunch of other engineers who came to biology to sort of make it into this engineering discipline. And so that's the language that they adopted, and basically one can plug in genes and various elements that control genes and design those genes to behave in a very predictable manner. If you want your gene to be highly expressed, right, or produce a lot of products and then drop off and produce less, you can do it. If you want an outcome that will be more or less linear, that's what you'll get. These are real experimental data derived from some of these engineered circuits of genes. So synthetic biologists think of genes as units and put them in circuits, genetic circuits. Let me give you one of the really cool examples of how far synthetic biology has come. So this is an example that deals with engineering of a baker's yeast, right? The yeast that produces beer or lets dough rise for bread. So the team of Jay Kisling has decided to make those yeast into a drug manufacturing facility. And the drug is artemisinin. This is a drug for malaria. Yeah, malaria is a pretty serious disease. It affects about 200 million people or so, if not more. About 1 million of them die, mostly children. We're talking about mostly, of course, Africa and perhaps Southeast Asia. Not so much a problem for the US, although we also have about 2,000 cases of malaria. And so the best drug to treat malaria is this molecule. And traditionally it's been made from the plant, Artemisia, that is either grown, and this you see here women in Tanzania collecting this plant. And so they would extract, not them, but I guess the pharmaceutical company will extract this compound, and it works remarkably well. In fact, that's a source for Nobel Prize for the Chinese scientist who has discovered this drug. But of course, growing these plants is not necessarily the most straightforward thing. And so Kisling and his colleagues decided to put genes from this plant and from other organisms into baker's yeast to make them produce this compound. Well, they couldn't quite go all the way, but what they have been able to do is they put all these genes, right? Each of these arrows represents a gene. They figured out how to regulate them properly, and they assembled this pathway of various genes whose product is not quite this drug, but something very close. And then subsequently one can do chemical modifications to arrive at this drug. And so now you have baker's yeast growing in vast vessels fairly cheaply, producing a precursor for this anti-malaria drug. And that's one of the mainstream synthetic biology endeavors, producing pharmaceuticals. Well, engineering yeast is not so difficult anymore and not so controversial. Engineering animals is more difficult and more controversial, of course. And so, but that's something that is very actively done in various labs in the world. And so I'll just give you a few examples of what is happening. So people are engineering fairly simple animals like these Drosophila flies to learn how various cell lineages develop into specific organs. Right, so I just give you one example of a fly which has a bunch of eyes on the tip of its legs. That's a weird monster. Well, the reason these researchers have done this is to learn how cell fate is determined. How from one cell you derive, well, in this case, an eye. And the reason they played with this is because, of course, this has direct consequences on our understanding of how to program various cells, including stem cells, to become certain tissues, right? So this is the path of learning how to make stem cells into potentially organs, say human organs, for transplants. Well, here's another example of engineered animal. This mouse is a mouse, except its immune system has been humanized. It has all immune cells from humans. Why would one do such a cruel thing to a little mouse? Well, the reason is that we do a lot of studies on mice. These are traditional lab animal, but mice are no small humans. They just aren't. And so what it turns out is that a lot of drugs that are developed based on mouse experiments, they don't work in humans. And in large part because immune system of humans is different. And so what people have done here by using genetic engineering is replacing mouse cells with human cells of immune system and making sure that these mice now have as close to an immune system of humans as one can, so one can do experiments with those mice and perhaps get more relevant data on how a human immune system would respond to certain treatments, whether it's cancer treatments or treatments of immune disorders. That becomes a very valuable engineered animal. Another really interesting development, not exactly in synthetic biology, but I guess in a very closely related field of cell engineering. These are embryos of pigs that have now liver cells from humans, and these are pigs that grow human livers. And the reason Craig Venter, who is doing these types of experiments, is doing this is because, of course, we have a huge shortage of human donors of various organs, and liver is one of those organs. And so he believes that sometime, at some point, probably very in the near future, these pigs will provide humanized livers that can be transplanted and not rejected by patients who need those transplants. He's done a lot of engineering on these pigs to make sure that they have no viruses that could otherwise jump from one kind of animal species to another. We'll see how that endeavor will work. So, what I was hoping to convince you in this first part of my talk is that engineering is something that is coming or has come to biology, that we know the parts, we have most of the tools, and of course the tools are being constantly perfected. We do understand the basic rules at least, and we have started developing a language of really programming complex engineering schemes in biology. Now, if you think about it, throughout human history, I mean, we've been doing biological engineering all along, right? For over 10,000 years, we've been domesticating animals and plants to the point where everything we eat has been genetically modified by us. It's just that we've been doing it blindly. We didn't know what to do, right? We let the nature select for the features that we wanted. And so doing it blindly, we've accomplished remarkable success. But I think that's where that success kind of ended, right? Because we didn't have the engineering tools. Now, perhaps with some more engineering tools, using natural selection, we've accomplished maybe this stage. And what is coming? That's exciting and amazing, right? And scary. Are we going to produce these types of elephants or these flying puglets? Probably not, right? I mean, scientists, for the most part, are on a very tight budget. So try proposing engineering something like this to a funding agency. That's a no-go. But there's a lot that we can do for medicine. So that concludes the first part of my talk, and if there are any questions, I'll be happy to answer them before I move on to tell you what my lab is doing in the realm of synthetic biology. And we're playing with light, and so I'll be talking a lot about light then. Please.
So do the scientists need to have an engineering degree as well as a biology degree?
The question is whether scientists need to have now engineering degree in addition to biology degree. That's right on target, absolutely. I happen to be a chemical engineer, so this is in my first degree, so this is not particularly foreign to me. At least the ideas are not foreign. They are indeed foreign to most biologists because the tools has just arrived, yeah, and they didn't arrive by design. As most things in science, real breakthroughs occur by chance. And so only recently we have gotten those tools to engineer precisely complex genomes like genomes of humans, for example, or other animals. Until very recently we couldn't do it. And so having some background in engineering is very helpful. Yeah? And in fact, if you look at the universities across the country, most major universities now have bioengineering degrees, departments, units, something like that, where they do teach both biology and engineering. Please.
With the program of CRISPR, can you tell us a little bit more about the engineering process? As I understand, in St. Jude's Hospital, they have been able to modify some of the genes for leukemia, and having 85% progress. Could you help us understand that CRISPR program?
Sure. Thank you. It's very nice to have such highly educated audience here. That's wonderful. I was afraid to even throw these terms in. So CRISPR-Cas indeed is one of the most advanced tools to precisely change genes, right? It's not the only tool, but it's the easiest one to use, and therefore in the last literally 4 or 5 years, probably the majority of labs that are involved in gene modifications have started to use this tool. Yeah? So what it allows to do is it allows a particular protein complex to recognize, basically scan this text of DNA, and with the help of a specific ribonucleotide, recognize where to stop and where to cut. Yeah, that's one of those tools that I was referring to. And so because it's very easy to, basically one can tell this complex where to go in this huge text that encodes genes, you have to go right there. So here's a little instruction for you, right? A little cheat sheet. Read it, go right there and do your cutting. And if we provide something to replace, say, a bad gene with, then that's what you do. So in a nutshell, but I'll be happy to give you more details, perhaps maybe at the break. So, but what the other part that you mentioned has to do with the progress in, I assume, immuno-oncology. Am I reading you correctly? I'm not exactly sure what study you have had in mind, but one of the, I guess, things in the news these days is indeed engineered immune cells, more precisely so-called T cells, or killer T cells. These are our immune cells that normally would recognize infected cells with bacteria or viruses or tumors and kill them. That's why they're called killer T cells. Problem with many tumors is that they become really smart and foxy. So they try to cloud the picture of those T cells. They try to evade them by all possible means, mostly telling other cells that they are fine. They're not supposed to be killed. And in fact, oftentimes recruiting those other types of T cells as guards of the tumors. Right? So they trick immune system at some point, and if they grow far enough, oftentimes they prevent that natural killing. So what people have used CRISPR technology for, and some of the other technologies similar to CRISPR, is to engineer T cells from a patient, take them from a patient, teach them ex vivo, or teach them in a dish basically, how to recognize those tumors better, right? How to ignore the signals that the tumor says, don't kill me, I'm good, right? So how to override that sort of tumor evasive techniques and engineer so-called recombinant receptors that will recognize those tumor cells regardless, whether they want it or not. Yeah, and so that's one of the indeed most promising ways of treating cancer. Teaching immune systems of humans to recognize tumors better. Yeah, I think it has incredible future. And in fact, some of the technologies that I am going to tell you about that we're developing in my lab might actually contribute to that direction as well. Because you need these cells not to be kind of mindless killers. You want to have some handle to tell them, well, it's time to kill, and at some point, all right, you've done your job, rest, or just disappear. Any other questions? Yes, by all means.
When you were introducing your talk, you mentioned that this whole topic of biological engineering is both exciting and scary. Now, so far in your talk, you've used the word exciting 3 times and the expression really cool 3 times. Are you going to get to the scary part?
Well, quite frankly, I was expecting the audience to bring the parts that scare you. Because as kind of a practitioner of the field, I have grown to appreciate the concerns that scientists usually have and the caution with which we approach these things. So from my perspective, it actually is not as scary when you understand the level of the safety nets that are built in these systems. But I'm sure that for the majority of this audience, these safety levels are just unknown. So what scares you? You tell me, and maybe I can address some of those concerns.
I need you to tell me.
Okay. All right. Well, what is scary? Well, I think the scary part is, of course, that we're dealing with kind of most fundamental issues here, right? And so if we change something in a human in such a way that it's irreversible, especially if it goes to the vertical line of descent, who knows what's going to happen, right? We don't have the luxury to experiment on humans like we do on mice. So we know precisely what happens to mice, right? We can keep them for a year. We can do it on multiple types of mice. We can get all the safety issues figured out with mice. Once we move to humans, they're unknowns. Yeah, and those unknown unknowns is where the scary part is in general. And therefore, what I guess my lab is trying to do is trying to design this extra level of safety, extra level of control, so that one doesn't quite change the animal itself, or the human in this case. We're working on engineering cells. And we think about those cells as being potentially part of human health as artificial organs that can be removed if necessary and can be controlled by either a patient or their doctor. Not exactly getting into the depth of that where you probably want me to go to, but maybe we can leave it for a round table discussion. Yes, Carl.
I'll answer your question that actually comes from literature, but it's from a novel by Naomi Mitchison. She was J.B.S. Haldane's sister, and he's one of the early figures in genetics in the 20th century. And she wrote a novel actually addressing not the human and animal implications, but the vegetable implications. And the book's called Not by Bread Alone. And there she's talking about sort of the cascade effects, where something gets adjusted, but then it has other genes expressed in many ways, just as you were talking about trying to define what they do. And in her novel, her projection, what happens is, something wonderful is engineered, it's solving food problems all over the world, and then, whatever its weakness is catches up with it, and there's sort of little food changes. And I'm wondering about how that one's even slightly different again, because in all the discussions of things like Monsanto, the question always is, well, plants pollinate, how do you stop that once you've got the genie out of the bottle? Runs around, but I think that would be certainly one of the fears that was expressed in the mid-20th century, in the 1960s, I think her novel is.
Thanks, Caroline, for bringing this issue of GMO plants and potential problems with those, right? So I try to stay away from plants just for the sake of time, but I'll try to briefly address it. We've lived as a humankind long enough to know that what we eat does affect us, but usually does not kill us unless we eat something really bad, and we almost always know what it is. We've eaten all sorts of plants, all sorts of animals throughout our history. They have had all sorts of microbes, and it's proven that our GI tract, our gastrointestinal tract, is a remarkable fermentor that will just grind everything, right? There's no example in human history that somebody would have died from a bad gene. One can die from toxin produced by plants or animals or bacteria, but it has never occurred that a gene would jump into humans. And we know it because we've sequenced many humans by now, right? They don't have any plant genes. So eating GMO things is safe. That is controversial in some circles, but that's the fact. So one may like it, one may dislike it, we can argue about it, we can discuss it more, but that's the reality of it, right? That's human history, it's long enough. A few thousand years is long enough. Now, the issue of contaminating other plants and spreading a feature that was designed for one particular plant, that is an important one. It comes to the issue of containment. And I think containment in any engineering processes is important. It's important to build a building that, if collapsed, will not kill people, right? It's important to build a building that if there's a fire, there's a plan of escape. So issues of containment and safety are applicable to all sorts of engineering, and I think, indeed, that's where we as bioengineers really need to work. Yes, please.
You know, kind of along with that, maybe you could very briefly talk about the difference between altering genomes just by human selection, for instance, at the beginning peas, selecting peas for different flower colors, as opposed to getting that same result by manipulating the genes directly. If you understand what I'm saying. Selection versus engineering.
I think I do. Which aspect of these two different approaches would you like me to address?
Well, compare them. Both of them end up with manipulating the gene structure, one in a more natural way, one in a more artificial way, engineered.
Right. Well, I mean, natural selection, right? Not natural selection, human selection of desired traits. I mean, basically, you're breeding, right? And you're getting something, and you don't know whether it works or not, right? In most cases, it doesn't. So you have to, kill those that don't work, and perhaps something works almost by chance, right? Which means not very high efficiency. And you keep going. As a result, you accumulate changes that are unpredictable and unknown, and that's why when you get to, say, pure breeds of dogs, oftentimes they have huge load of genetic disorders because nobody has selected against those, right? Because you start from a healthy, so to speak, individual, and then you accumulate the traits that you desire and the traits that you don't. You do it blindly. And so the difference is that, well, if you do know what to change and you do know how to change it, it makes perfect sense to change it precisely, right? And in fact, this has started to be one of the human gene therapies, right? So there are several diseases that are affected by a single mutation in a gene, and those genes are known. And there are companies that are changing cells in a very specific manner, just restoring things to normalcy. And so, say people with cell sickle anemia don't have anemia anymore. They can become normal if some of their cells are replaced with normal cells. They are cells that have been engineered to not have that deleterious mutation. Well, for the sake of time, maybe I can give you some glimpse of what my lab is doing, because I'm actually very excited to share it with you, and then we can continue with the questions at the end. So, this is, I guess, one of the major questions that my lab is working on. How can we teach a cell, not a smart dog like this, not a smart German Shepherd, to respond to a command? We want to command cells. We want to control cells. If we engineer them and put them in, say, in a human, we want to be able to have external control. We want to tell them, work now, don't work later, work here, don't work there. How do you do that? How do you talk to cells? Right? If we talk to animals, you can rely on their sense of hearing or sight. Cells, they don't have ears, and most cells don't have eyes either. So what my lab has been doing for the last few years is kind of engineering eyes to human cells. That may sound bizarre, but in essence, that's what we've been trying to do. So I don't know how to engineer ears to cells, but I think we have some ideas to how to engineer eyes so that one can make a command with light to cells and turn this smiley little human cell into a monster that would, say, attack a tumor. It's a basic issue of domestication, right? So sometimes I think about it as an issue of domesticating cells, right? Because domestication is basically making animals respond in a predictable fashion to human commands, right? That's how we domesticate animals. Well, can we do it with cells? And so, like I said, I think we can do it with light. And let me try to explain to you what I mean by that. Because most human cells, except for cells in our eyes, some cells in our brain, they don't really respond to light. They don't know what light is. We're kind of blind. And of course, we're not really working with humans, we're working with mice. There's one particular type of light that we're interested in, and that is red and near-infrared light. And the reason we're interested in this type of light is quite simple, because that is the light that goes through human tissues. And you have examples here of a fist. You can see that this fist holds some sort of red light, and that red light goes right through. This is human breast, and you can see that there's enough light that this breast tissue is all red. And I encourage you to play with your cell phones. Well, those of you who are most adventurous, pick up your cell phone, turn the flashlight function, right, and put it, I don't know, to your thumb. You see it's red? It's not blue. It's not green, it's red. And that's just the nature of blood essentially absorbing most of the light, and most of the light not being dispersed very easily, but not quite red. And so that's why we're interested in teaching human cells to see red light. And red light is very safe. It's been used in various types of medical applications for decades, so we know if we can do it, it's going to be a safe technology. Now, how do we teach human cells to see light? Well, we go to the parts, and the best parts for seeing light are in bacteria. In bacteria, there are photoreceptors called bacteriophytochromes. They absorb light in the farthest red region compared to all other types of photoreceptors. These are the photoreceptors that we have in our eyes. They allow us to see from blue to red, but not quite as far red as some of these bacteria can see. And so we take these parts, and this is one of those proteins in bacteria. You can see that I'm showing it as a composite element, so it has these blocks. These are protein modules. And in this case, we're engineering a new protein module that will sense light. So this is a protein that naturally senses light in a bacterium called Rhodobacter. And we want to make a different protein with different activity that we think is going to be very important for controlling neurons and for controlling other human cells. And so we take this module called Adenylate cyclase and basically try to do this new fusion. So essentially we're Legoing in this case, not really truly engineering, because the engineering rules here are not quite known. We have deciphered some of them, but it's still a bit fuzzy. But in essence, since we know the blocks from which this protein is made, we can substitute some of the blocks, so essentially changing this red roof on this house with a blue roof. Still a house. It turns out that with some ingenuity and a lot of lab work, one can accomplish this feat, right? It took us maybe 3 years to do it, but that has been done. Once we did it, we realized that this protein works great in bacteria, but doesn't work great in humans, because it just is made from blocks that are not designed to not designed to work at human temperature. So we went on the hunt for other parts that will work better at body temperature. And sure enough, the part from a human pathogen called Mycobacterium tuberculosis, right, that part works fine at human temperature. It's a pathogen, no surprise. And so we made a new chimeric protein, and we replaced the original photosensory module from another one, from a bacterium called Deinococcus. And so you can see this is a bacterial soup right there. Rhodobacter, Nostoc, Mycobacterium, Deinococcus, these are all bacteria from which we're taking those parts and making them to work. And so one of the applications of this particular protein that I showed you is in actually studying how our long-term memories are formed. That's a long jump. From bacteria to memories, how is it all connected? Well, it's connected because the protein that we made controls certain ion channels that operate in neurons in mice and humans. And some of these neurons are involved in connecting thalamus to cortex in the brain. And believe it or not, when we sleep, these two regions of brain exchange electrical signals that are sent as these wavy features. They're called spindle oscillations. And these spindle oscillations are controlled by the molecule that our protein makes. And so the reason these two regions of brain are talking to each other during sleep is to establish some of the memories, some of the short-term memories, survival long-term. Not all memories are deemed to survive, but some do, and that's the process with which these two different brain regions talk to each other. And so that's what people have hypothesized, but they could never really understand whether that's true or not. Well, with the tool that we've designed, it now becomes possible. And so our colleague at the University of Wyoming, Dr. Kin Kwan Sun, in zoology department, he has placed our gene in the brains of mice. This is a mouse. It's irradiated by red light. Unfortunately, mice don't really see red light, so we don't disturb their sleep. What we can do, though, we can see that these oscillations can be repressed, can be inhibited when we turn this red light on. So now we have a tool to see whether we can really understand how long-term memories are formed in mice. Right? So very unusual application. Well, here's another application that we're thinking about. So since we made this interesting protein that responds to red light, maybe we can design a way to treat an illness like diabetes. Right? Diabetes or a variety of other diseases that are due to the lack of specific hormones. Hormones are really easy to make, right? If we can make them under the control of red light, we basically can have a cell that is engineered to produce those hormones, and that cell becomes a drug factory itself. If we can implant those cells in a human, in perhaps a contained manner, all it will take to turn production of those hormones will be light. So imagine that type 1 diabetics who can turn light on their synthetic organ with those cells and produce a dose of insulin that they need. I'm more excited about other applications, like maybe applications to people like myself who are not quite obese, but on the overweight side, because there are hormones that control appetite. So if I could have these cells that would produce anti-appetite or anti-satiety hormones, I could actually, enjoy my food, but not quite as much of it. And so I think this would be another potential application of it. Well, here's another potential application, right? And that deals with immune suppression. Soldiers come from the wars where our not necessarily smart politicians send our soldiers to, and they come without legs or arms. And one of the best ways of replacing them is actually finding a donor and putting another arm. And remarkably, the surgeons can do it. What they can't do is they cannot teach immune system to not reject that arm or leg after a period of time. So after a period of time, almost inevitably, they have to be removed. That's a travesty. What we're trying to do is we're trying to engineer localized immunosuppression, which means we'll have immune cells that respond to light in a particular organ, not everywhere. Because right now, that's the standard of treatment. People have full body immunosuppression, which makes them extremely susceptible to all sorts of diseases. Any cold virus becomes a life-threatening disease. So what we're trying to do is we're trying to make these light controls immune cells that will prevent rejection locally. Well, on that note, I guess I'd like to tell you that basically what we're doing is we're engineering cells to perform different functions, some nasty, some really good, right? And so that's the essence of synthetic biology and biorevolution, and I encourage you not to be too afraid of it. And I'm out of time, so I guess we'll have questions on this part, perhaps at the round table. Thank you so much for your attention.